Well, howdy there, folks. I'm going to teach you about tissue recellularization in 3 minutes or less on this fine afternoon. Let's go. Tissue recellularization is basically the process of taking an organ or organism, stripping it of all of its cells, and then refilling it with the cells of a different organism.
Wait, I thought living things were made entirely of cells. What's left after you remove them?
Cells actually reside within a structure called an extracellular matrix, or ECM for short. It's a sturdy web of proteins and carbohydrates. In plants, it's made of cellulose, and in animals, it's usually collagen. For instance, our bones are parts of our living bodies. They contain plenty of cells, but bones can be bleached, cleaned, dried, and we're left behind with a sturdy structure of calcium phosphate and collagen.
All right, I follow. Proceed.
Back to the topic at hand. Before we recellularize any tissues, we're first going to have to decellularize them by destroying and rinsing the cells from the matrix without damaging the ECM. There are plenty of substances we can use to do this, each with their own pros and cons.
Namely acids and bases, hypertonic and hypotonic solutions, detergents, and enzyme solutions.
We're finished with the decellularization process when the DNA content of our tissue is 50 nanograms per microgram. We're left with the extracellular matrix, which is a cool translucent white ghost of our original tissue.
What now?
Now we fill her up. The process of using stem cells to repopulate a scaffold is called seeding. There are two types generally, static and dynamic seeding. For the sake of time, we're just going to focus on static seeding. Basically, we would need to mix up a broth where the stem cells can chill and get all their nutrients while they wait for further instruction, like a living homogeneous meat soup.
That's pretty gross.
Ugh, I know, right? Anyway, this next step is pretty simple. You submerge your ghost tissue in your meat broth, let it chill for several days, the stem cells will do their thing, and then presto, you've got some freaks of nature fresh off the griddle. Here's a great made of meat. This one's spinachly full of living human cells.
But why? Just to say you made meat berries in human salad?
That's the awesome thing about this new emerging field of research. Two very exciting future applications: organ transplants and slaughter-free meat.
Transplant patients can wait years to receive donor organs, and even then they have to take anti-rejection drugs for the rest of their lives. If we could synthesize an organ from the recipient's very own stem cells, we could perform transplants on command with the patient's own cells, eliminating the need for immunosuppressants. In fact, heart valve transplants regularly occur with recellularized valves from pig hearts.
The other exciting prospect of tissue recellularization is slaughter-free meat.
We could decellularize plants or fungi like oyster mushrooms or jackfruit and fill it back up with stem cells from cows, pigs, chickens, alligators, whatever floats your boat. And since stem cell harvesting is hardly more invasive than getting blood drawn, no animals would have to die for your meat fruit fillet.
Whoa, okay, awesome. So why isn't meat fruit at the grocery store right now?
Murphy's Law, baby. Nothing is as easy as it seems, everything takes longer than you expect, and if something can go wrong, it will. That being said, it is certainly a possibility in the future. And on that note, you could ethically try human meat.
Whoa, what? Don't take it there, that's weird.
I'm just saying, I'm not